Hey, it's time to try comparing shifts in distributions and allele frequencies. Let's go. It's me, Moser. After this video, you should be able to distinguish between graphic representations of distributions and of allele frequencies. You should be able to compare distributions from populations that are taken at different times to determine if evolution has occurred. And you should be able to compare allele frequencies from a population taken at different times to determine, again, if evolution has occurred. It'd be just dandy if you knew what distributions and allele frequencies were. Hey, remember evolution? Well, let's recall the three definitions of evolution that we learned in Level 1. The first one, the broadest and simplest, is biological change in a species over time. The second is the change in the distribution of a trait in a population over generations. And the third is the change in the frequency of an allele in a population over generations. So we're going to focus on those last two, a change in the distribution of a trait in a population and the change in the frequency of an allele in a population. And we're going to look at how we can show them and compare information about them. So a distribution, you remember, is how a trait is distributed in a population. And the normal distribution that's shown here is pretty common. All it means is that the largest number of individuals for any particular trait that have the particular measurement are going to be found towards the middle of the range of the values for that trait. Pretty common, really. So how do we get a distribution figured out? Well, we generally don't have the ability to go out and measure the ears or the tail length or the foot size or whatever the trait is in an entire population. So we measure a number of individuals, as large a number as we can, chosen at random, and assume that the sample, that's what we call that smaller group that we measure, is a good representation of a population. Here we've got pictures of some biologists taking sample data from wildlife populations. So we're weighing fish, we're measuring turtle shells, we're measuring the nose on an adorable baby bear. Oh my goodness, I want that job. Of course, allele frequency is a little bit different, where distributions deal with phenotype, allele frequencies deal with genotype. And an allele frequency is how common an allele is in the population, or what percentage of the total alleles in a population are the allele of interest. And it looks pretty much like this. Again, we don't go out and take genetic information from every individual in a population. That just wouldn't be possible. So we sample the population in some way that allows us to choose the individuals we sample randomly. In the case of figuring allele frequencies, take actual biological material that we can get DNA out of from those individuals. Here we've got three instances of biologists taking DNA from wildlife. In this case, we have a wildlife fisheries biologist who is pulling scale samples from a large fish. Here we have a group of individuals working with a sedated large cat, and they're not only collaring it, but they're drawing a blood sample to get DNA. And here we have maybe the most interesting. This is a trap that's designed not to trap the animal, but to trap a piece of their fur. So it's used with predators, and you hang some sort of bait, some sort of nice, juicy, fresh meat in such a way that to get to the meat, they have to go past something that's going to catch a little piece of their hair. Because in fact, you can pull DNA from that hair. Pretty clever. So anyway, again, we sample the population, and then we can graph allele frequency. Meaning, we figure out what percentage of the population is carrying a particular allele, or what percentage of all the alleles that we find are the particular allele of interest. Now, each of these is just a bunch of numeric data, really, and it doesn't make a whole lot of sense until we can organize that data in some way that allows us to see it clearly. So, how do these actually look on paper or on screen? Well, you should remember this. This is, again, a normal distribution. And here we're looking at ear length in Canis latrans. Canis latrans is the coyote. And this is information data from 1986. Doesn't say where. I'm sure that information is somewhere out there. And what we see is that the most common number, the most common measurements, are somewhere here in the 14 to 15 centimeter range for ear length. Okay, we have some individuals who are down here on the range of 7 centimeter ears, little stumpy ears, and some who are up here in the range of 23 centimeter ears, really big, long ears. But that's the distribution. Of course, allele frequencies look different because we're not doing the exact same thing. Here, we're looking at what percentage of all the alleles in a population are either the dominant or the recessive allele. Note that because we can only, every individual is going to have one or the other or both. So the total number of alleles for this gene have to add up to 100, meaning if we have a 0.25 for this one, we got to have a 0.75 for this one. It always has to add up to 1. Okay, great. So that's what a normal distribution and an allele frequency chart look like, but wait, we're interested in seeing if these things are changing because that's the definition of evolution, and that's what we're here to talk about. So check this out. This is a distribution showing the mass of female walleye in some particular lake somewhere in Ohio, and this is data from 1964. Okay, great. What do we see? Well, we see that the mode of the data, the highest bar, the measurement that has the most number of individuals that have that measurement, is about 9 kilograms. That's a nice sized fish. That's about an 18-pound walleye. That's a good fish. Now, interestingly enough, we have more recent data, not totally updated, but more recent at least. We have mass of female walleye from that same body of water from 1999. And what do you see? Well, the mode for this data is approximately 7 kilograms. Wait, I thought it was 9 before. Oh yeah, it was. So when we compare these two distributions side by side and ask, has there been a change in the average mass of female walleyes in the same age range in the same lake over the last 25 years, 35 years? Well, yeah, there has been. The female walleye population seems to have gotten smaller. Has evolution happened? Yes, evolution has happened right here. It's not that complicated. Excellent. So let's look at a particular gene found in walleye, the C sub 3 gene, and we have the dominant C sub 3 and the recessive small c sub 3. This gene controls some enzyme in fish. Don't know what and don't need to. But biologists had taken sample data from walleye in a particular lake in 2009, and every 2 years they were able to obtain genetic samples from these fish, and they could chart the frequency of the big C3 versus the little C3 in walleye. Well, what do we see? We see that starting off in 2009, the dominant allele was made up just less than 50% of all the alleles in the population, and the recessive allele made up a little bit more than 50%. Every year, the percentage of alleles made up by the recessive, the small c3, dropped until we get to 2019, when almost 100% of the alleles in the population are the big C3, and the little C3 has almost completely disappeared. Guess what? Evolution happened here. With regards to this gene, this walleye population has in fact evolved. Now, each of these examples is a very narrow picture of the kind of changes that can occur in a species or a population. Evolution in size or evolution in a single gene. But very often we want to see a larger picture. So we might want to look at several traits at once, such as this. So here we're looking at distribution of phenotypes in Ursus americanus. That's the black bear. We've measured, or biologists have measured, over the course of some number of years, skull width, leg length, the distance between their eyes, inner eye distance, and tail length. And have any of these traits shifted their distribution? Well, yeah, they all have. So this is really serious change. Okay, evolution has happened here. Here we've got allele frequency charts for 4 different genes, big B little b, big N little n, big Q little q, and big G little g, in Odocoilus virginianus. That'd be the white-tailed deer to you and I. Now, if we look at the first 2 genes on the top, there really hasn't been a lot of change in the frequency of those 2 genes in the dominant and recessive alleles. On the Q versus Q, there hasn't been a lot of change. They've drifted up and down a little bit. There's been some change on the big G versus little g, but not a lot. So is this population perfectly stable? No. There seems to be some shift with regards to this gene, but overall, this population is relatively stable. And data like this can show us either a population that is changing or a population that's staying mostly the same. All right, let's review the learning objectives. Can you distinguish between a graphic representation of a distribution versus an allele frequency? Can you compare distributions from a population that are taken at different times to determine if evolution has occurred? And can you compare allele frequencies from populations taken at different times? Again, just to determine if evolution has occurred? Well, I hope you can. If you have questions, let me know.